Dear student, I am Dr. Prakash Pigli, Professor of Biochemistry. So in this lecture, I will be going over classification of lipids and applied clinical aspect. So in my previous lecture, I have explained you classification of fatty acids, different types of fatty acids. So in this lecture, I will be going over classification of lipids, which are little more complex than the fatty acids. Because lipids have more and more fatty acids being attached along with other molecules like glycerol, phosphate, carbohydrate, which are being attached to the fatty acids. Okay, so that type of lipids we are going to classify now. So lipids are simply classified into simple lipids. So simple lipids are the ones when you break them or when you hydrolyze simple lipids, you get fatty acids. Generally, lipid molecules you're going to get. And the compound lipids are the ones that along with the fatty acid, along with the lipid material in the compound lipids, we also get carbohydrates or nitrogen, nitrogenous base, phosphates. This kind of other molecules you're going to get from compound lipids. And the derived lipids are the lipids that are derived from simple lipids are the fatty acids molecules like cholesterol is a derived lipid or bile acids is a derived lipid, steroid hormones is you can put them as a derived lipid. So this is how simply we can classify lipids into simple lipids, compound lipids and derived lipids. Now let's look at some of the more examples in simple lipids, compound lipids and derived lipids. First let's look into the simple lipids. Simple lipids are basically these are esters of fatty acids with various alcohols that are being attached with. Like example, triacylglycerol here. Triacylglycerol, it is a glycerol which has got 3 carbons attached with a fatty acid. Glycerol attached with the 3 fatty acids is a triacylglycerol molecule. So that's basically a type of simple lipid. And then so waxes. So fats and waxes are example for simple lipids. So what are fats here? So fats are fatty acids with a glycerol molecule. Example triacylglycerol, which is stored in the adipose tissue in human body. Oils are these are fats in liquid state. Example like sunflower oil, safflower oil. So basically they contain triacylglycerol but the liquid in the liquid form. Coming to waxes, which are also simple lipids. So waxes are these are esters of unsaturated fatty acids with a long chain alcohol, and the unsaturation or the length of carbon of unsaturated fatty acid can be 14 to 36 carbon, like long chain to very long chain fatty acids, and fatty acid or long chain alcohols, it can go from 16 to 22 carbons. So in humans and animals, waxes are secreted by skin glands and it helps to keep skin lubricated, waterproof and also pliable. So that's the use of waxes. Okay, so waxes and fatty acids are example for simple lipids and that you must remember. Coming to compound lipids. Compound lipids, depending on the additional other than the lipid molecule, the molecule that is present in compound lipids, it can be phosphate or it can be carbohydrate. If the phosphate containing lipids are there, we refer that as phospholipid. If the carbohydrate containing lipid is there, we just simply refer that as glycolipid. Now, what are the phospholipids we have? One of the example for phospholipid, we have phosphoglycerides. We are going to go into more examples on phosphoglycerides. And then we have sphingomyelin. So, both phosphoglyceride and sphingomyelin are the example for phospholipid. Coming to glycolipids, glycolipids can be cerebrosides, sulfatides, globosides and gangliosides. We are going to go into details of all of them, cerebrosides, sulfatides, then globosides and gangliosides, each one of them, and also we are going to get into the examples. Now, the compound lipids, these are esters of fatty acids containing groups in addition to alcohol and one or more fatty acid. So, as I said in the earlier slide, we can classify compound lipids as phospholipids and glycolipids. Phospholipids, they contain phosphoric acid and other substances in addition to the fatty acid and alcohol. Example for phospholipid, again we can classify based on the backbone present in the phospholipid. If the glycerol backbone is there in the phospholipid, we call that as a glycerophospholipid. If the backbone is sphingosine, we call that as sphingophospholipid. Okay, so glycolipids, they contain carbohydrate, which is sometimes referred as glycosphingolipids. So they contain sphingosine, fatty acid and N-acetylated carbohydrates. Sometimes these N-acetylated carbohydrates can be sulfated. Sulfur can be attached to this carbohydrate molecule. So we have cerebrosides, sulfatides, globosides and gangliosides as example for glycolipids. And then other complex lipids, these are associated with lipids associated with proteins. In that we can actually put lipoproteins. What are the lipoproteins? We have HDL, LDL, VLDL and then chylomicrons. So these are all lipoproteins. Basically these are combination of proteins plus a lipid. Now within that classification of lipids, so as we have already seen, simple lipids, we have triglycerides and waxes. Component lipids, we have phospholipids. Within that we have seen phosphoglycerides and sphingomyelin. So phosphoglycerides, we have say glycerol as a backbone, glycerophospholipids, or sphingomyelin, sphingosine as the backbone, sphingolipids there. Now in the glycolipids, as we have already seen, so we have cerebrosides, sulfatides, globosides and gangliosides. So derived lipids, we have a fatty acid which is saturated or unsaturated fatty acid steroids and their fatty acid esters, like example quinones, ketone bodies, lipoproteins, polyisoprenoid compounds, they can be part of this. Now cholesterol and cholesterol esters, triacylglycerols, these are all termed as a neutral lipids because they are uncharged under physiological pH. You need to take a note of that. Now, this is another way of classifying lipids here. So, we have storage lipid and we can broadly classify them into membrane lipids depending on what is the function of these lipid molecules and where exactly they are found. So, storage lipid, most abundant storage lipid for our body is triacylglycerol, which is stored in the adipose tissue. As you see here, it is a glycerol backbone with fatty acid attached, 3 fatty acids attached. Whereas membrane lipids further classified into phospholipids and glycolipids. Phospholipids further classified into glycerophospholipids and sphingophospholipid. So, and then the glycolipids can be sphingolipid. As you see here, sphingosine is the backbone, fatty acid is there, along with that monosaccharide or di or oligosaccharide. This is the carbohydrate component will be present in glycolipids. Now, let's look into triacylglycerol, which is a simple lipid, which is a storage lipid for us. Triacylglycerol, as I already said, it is an ester of glycerol with 3 fatty acids attached. The structure is shown here. So, here is the 3 carbon molecule. So, we have number 1, 2, 3 carbon there, and fatty acid is attached to those 3 carbon molecule as an ester bond, as you see here. The O, this is the alcohol group of glycerol, and that OH is taken out and fatty acid is attached here, R1, R2 and R3. So, these fatty acids can be of varying length. It can be short chain, medium chain, long chain fatty acids, very long chain fatty acid. These fatty acids can be saturated or unsaturated fatty acid. So, any type of fatty acids can be attached to these 3 carbons here. So, generally speaking, out of 3 fatty acids that are attached to triacylglycerol, 2 of them will be unsaturated fatty acid and 1 will be saturated fatty acid in a triacylglycerol. So, that means 2/3 of our body fat, it contains unsaturated fatty acid, because out of these 3 fatty acids attached to a triacylglycerol, 2 out of 3 will be unsaturated fatty acid. So, because triacylglycerol is a storage fat in our adipose tissue. So, 2/3 of our body fat actually has unsaturated fatty acid. Remember that. So, examples of foods that are containing triacylglycerols like oils, butter and other food fats, they are rich in triacylglycerol. Remember that. Now, triacylglycerol is completely non-polar as it does not have any polar groups, because the hydroxyl group of glycerol, glycerol is it contains 3 alcohol groups OH, and that OH is taken out the H is taken out and fatty acids are added to all the 3 carbons. So there is no polar group here in the triacylglycerol molecule. So it's a completely unpolar molecule or non-polar molecule. And also note that it is simplest and most abundant lipid of all the lipids. Triacylglycerol is the most simplest and also the most abundant lipid that is present in our body, and it is sometimes referred as just a fat or a neutral fat or triglyceride. Instead of saying it as triacylglycerol, we can simply call it as fat or a neutral fat or a just triglyceride. Okay, so it is the major storage form of fat in our body, stored in the adipose tissue, which can meet the energy needs of our body for several weeks when we go into fasting and starvation. You got to be aware of that. And also, as such, in terms of calories, so fatty acid oxidation, fatty acids, it gives 9 kilocalories per gram of fat. Okay, remember that. So which is the highest compared to carbohydrates and proteins. Remember that point. So that's why our body tried to store fatty acid as the storage form of energy in the form of triacylglycerol. So what are the functions of triacylglycerol here? So as I was saying, it is the storage form of chemical energy that is stored in our body almost in an unlimited quantities, because triacylglycerol, unlimited quantity of triacylglycerol can be stored in the adipose tissue for any needs in future. Just in case if the person goes into fasting and starvation, so this triacylglycerol can be broken down and it can give energy. And also triacylglycerol in the adipose tissue, especially present in subcutaneous tissue, it can act as a thermal insulator against cold temperature or cold atmosphere, because in response to cold there will be fatty acid lipolysis, and that will, especially the long chain fatty acids, it can lead to opening of thermogenin or the uncoupling proteins, and that will lead to increased generation of heat. So that is one of the way a thermal insulator effect can be done. And also as such, the presence of fat in the adipose tissue will protect our the tissue beneath the adipose tissue kind of insulated from the cold temperature. Mechanical, and also it can act as a mechanical shock absorber, especially if fatty acids means adipose tissue or the fat that is around several organs which is present there, so that will kind of give a cushioning to visceral organs, so giving a mechanical shock absorber function to it. Coming to phospholipid. Now as you see here, the phospholipids, these are basically the phosphatidic acid, number 1, 2, 3 carbon, that's a glycerol, and then we have carbon number 1 attached with the fatty acid, carbon number 2 attached with the fatty acid, carbon number 3 attached with the phosphate. That's what is the phospholipid formation here. So basic structure of phospholipid formation is phosphatidic acid. So what is phosphatidic acid? It's a glycerol backbone having number 1, 2, 3 carbon, first and second carbon attached with fatty acid, and the third carbon is attached with phosphate. Okay. And now if you remove that phosphate and add with or if you add some other functional group or some other group to phosphate, so you get a different types of phospholipids there. So phosphoglycerides it will be like this. We have number 1, 2, 3 carbon there, and first carbon fatty acid, second carbon fatty acid, third carbon phosphate, to that a polar alcohol group attached, like if it is attached with OH or something here. So that is phosphoglyceride. So this phosphoglyceride can be used as a basic unit and make different types of phospholipids. Let me show you that in the table later on. So phospholipids they are amphipathic molecules because they have got phosphate attached, which is a PO4 minus, and that phosphate gives a polarity. And also phospholipids are most abundant lipids in biological membrane, like more than 50% of membrane lipids are phospholipids. Remember that. And two types of phospholipids depending on the backbone. That is, if the glycerol is the backbone where fatty acids and phosphate is attached, that's called as glycerophospholipid. If the sphingosine is the backbone where fatty acid and phosphate is attached, that is called as sphingospholipids. Now, glycerophospholipids are all derivatives of phosphatidic acid, and the common alcohol groups of phosphoglycerides are either ethanolamine, choline, amino acid serine, myoinositol and glycerols. Basically, in this alcohol group, if ethanolamine is added or myoinositol is added, so or any other functional group attached to that, so you get a different types of glycerophospholipids. So here in this table you can understand, so if this OH is replaced by ethanolamine, okay, so basically this OH group here, okay, if this OH group is replaced by ethanolamine, so you get phosphatidyl ethanolamine. Like that, if it is replaced by choline, so we get phosphatidyl choline. And note that phosphatidyl choline is also referred as lecithin, and it is the most abundant type of phospholipid present in our cell membrane. We are going to go into more details on phosphatidyl choline, or which is also called as lecithin, later. Now, if a serine is replacing there, so we call it phosphatidyl serine. If myoinositol is replacing group there, we call it as phosphatidyl inositol. If a glycerol is replacing to the OH group, it is phosphatidyl glycerol. And if phosphatidyl glycerol is replacing that OH group, it is referred as cardiolipin, which is also diphosphatidyl glycerol molecule. So, depending on the replacement group attached to that phosphate in a phospholipid molecule, so we get a different types of phospholipids. So, these are examples for phospholipids. So, one more time. So, I am just giving you the examples of phospholipids. So, we have phosphatidyl ethanolamine, phosphatidyl choline, which is called as lecithin, which is the most abundant component of something called as surfactant in the lung alveoli. Then we have phosphatidyl serine, phosphatidyl inositol, then phosphatidyl glycerol and cardiolipin, which is a diphosphatidyl glycerol. And one of the things that I would like to mention with the phosphatidyl inositol is there is something called as PIP2. PIP2 is phosphatidyl inositol 4,5-bisphosphate. Phosphatidyl inositol 4,5-bisphosphate is what? PIP2, which is a membrane phospholipid, and whenever this membrane phospholipid is hydrolyzed, you are going to get a DAG, that is diacylglycerol, and IP3, inositol triphosphate. These are second messengers. Okay, so that type of lipids can be derived from a membrane lipid. Now let's go into more on phosphatidyl choline, which is also called as lecithin. So it is a glycerophospholipid containing a choline molecule. In fact, it is a storage form of choline in our membrane. So it is the most abundant phospholipid present in our cell membrane. Make sure you remember that phosphatidyl choline is the most abundant type of phospholipid present in the cell membrane. And also it is the most abundant body store of choline, because if you look at the structure of phosphatidyl choline, here is the structure of phosphatidyl choline. It has one fatty acid attached to the first carbon, second fatty acid attached to the second carbon, third carbon with the phosphate, and to that phosphate choline is attached. This entire structure here is choline. Okay, so as you see, choline has 3 methyl groups which can be donated. So the storage form of choline in our body, especially present in the membrane, is phosphatidyl choline or lecithin. Okay. Now, what is the use of choline? Choline can be used to synthesize a parasympathetic neurotransmitter, that is acetylcholine. So, whenever there is a need for synthesizing acetylcholine in parasympathetic neurotransmitter, choline from here can be taken. It is attached with acetyl-CoA, and that makes acetylcholine. CoA is released and acetylcholine is made. Parasympathetic neurotransmitter is made. And also phosphatidyl choline, because it contains choline there with methyl groups, these methyl groups can be donated. So it can act as a methyl group donor also. Remember that. Now, where exactly you see highest concentration of phosphatidyl choline or lecithin? So, the type of phosphatidyl choline which is found in abundant concentration in the surfactant which is synthesized and secreted by alveolar cells, that is dipalmityl phosphatidyl choline, which is also referred as dipalmityl lecithin. So, as it is shown in the figure here, see the alveoli, it is lined by a surfactant. See, there is a surfactant layer here, and this surfactant which is coating the inner layer of the alveoli, it will prevent collapse of the alveoli. Whenever exhalation is done, the alveoli can collapse, and that is prevented by having a surfactant coating in the inner layer of the alveoli. So, the type of cells that will synthesize and secrete surfactant is alveolar type 2 cells, that is type 2 pneumocytes. There can be type 1, type 2 pneumocytes in the alveoli. Type 2 pneumocytes are the ones that will synthesize and secrete surfactant. Generally, surfactant synthesis, it starts 24 to 28 weeks of gestation, and good quantity of surfactant can be found in the amniotic fluid by 28 weeks of gestation, 28 to 32 weeks of gestation, and by 34 weeks of gestation and onwards. So, there will be significant quantity of surfactant being synthesized. So, you got to remember that point here. So the dipalmityl phosphatidylcholine, it is a type of phosphatidylcholine with C1 and C2 attached with palmitic acid and C3 attached with the choline molecule. So, that structure is shown here. As you see, number 1, number 2, number 3 carbon. Number 1 carbon, there is a palmitic acid. This is a 16 carbon fatty acid. Number 2 carbon also attached with the palmitic acid. That's why it is dipalmityl. Okay. And then the third carbon is attached with the phosphate, and to that phosphate choline is attached. That's why it is called as dipalmityl phosphatidylcholine, which is also referred as dipalmityl lecithin, because lecithin is nothing but phosphatidylcholine. So, that's the structural aspect there. Now, it is an important component of surfactant, and it is synthesized by type 2 pneumocytes, and it is going to lower the surface tension of alveoli, increases alveolar compliance, decreases the effort to expand the alveoli, and thereby it prevents alveolar collapse during exhalation process. That's very important function of surfactant. So, that means surfactant should be sufficiently available to prevent alveolar collapse. Now, the surfactant synthesis, it begins 24 to 28 weeks of pregnancy, and significant quantities of surfactant will start appearing in the amniotic fluid by 28 to 32 weeks of gestation, and by 35 weeks of gestation, most babies will have adequate amounts of surfactant. Now, what if there is a decrease in the surfactant synthesis? If there is a decrease in the surfactant synthesis, it will give rise to a disease called as infant respiratory distress syndrome. So, this infant respiratory distress syndrome is also called as neonatal respiratory distress syndrome or hyaline membrane disease. So, what happens here is neonates, especially premature infants, whenever the birth occurs before 35 weeks of gestation, so usually they will have insufficient surfactant being synthesized, and that will lead to collapse of the alveoli, because surfactant prevents alveolar collapse. Insufficient surfactant means alveoli will be collapsed after exhalation, and it will be difficult to open them without much of surfactant. So, that means when inhalation is done, so they still remain collapsed. So, that will lead to decreased oxygenation status in the body, and that will give rise to signs and symptoms. So, signs and symptoms of infant respiratory distress syndrome begin shortly after birth, and some of the important clinical signs that you need to expect here is because of lack of oxygenation. So, there will be tachypnea, increase in the rate of respiration, tachycardia, increase in the heart rate, chest wall retraction, expiratory grunting, flaring of the nostril, cyanosis because of lack of oxygenation process. And also when the X-ray of chest is taken, because alveoli are collapsed, there is no air filling going on here sufficiently. So, you don't see a clear lung field. So, you are going to see something like this. So, there is no clear lung field here. So, because of the collapse of the alveoli. So, that type of feature we call it as ground glass appearance in the chest X-ray. Make sure you remember that important point. Now, what are the other glycerophospholipids? Other glycerophospholipids, we have phosphatidylethanolammine, which is called as cephalin, phosphatidylserine. So, they are part of our cell membrane. And the phosphatidylserine also plays a role in apoptosis or programmed cell death. And phosphatidylinositol being present in cell membrane, it plays an important role in cell signaling and membrane trafficking process, because phosphatidylinositol 4,5-bisphosphate, which is present over cell membrane, when it is clipped, as I told you earlier, it is going to give diacylglycerol and inositol triphosphate. Both diacylglycerol and inositol triphosphate, they are second messengers. So, that's how they participate in cell signaling process. Now, there is something called as plasmalogens. Plasmalogens, which are about like 10 to 30% of phospholipids, which are seen in brain and heart. These are plasmalogens, and plasmalogens, they are either having a ether link on the first carbon instead of ester linkage found in the triacylglycerol. Let me show you that. So the first carbon, we have a ether linkage here. Okay. So compared to what we have seen with the second carbon where fatty acid is added. So the first carbon fatty acid is attached with the ether linkage rather than the ester linkage in a plasmalogen, unlike what we see in triacylglycerol. And also, of course, there will be phosphate attached to the third carbon, and to that phosphate ethanolamine is attached. So this is what is the structure of plasmalogen. So basically it has two fatty acids, a phosphate that is attached to the third carbon of glycerol, and additionally there will be ethanolamine that is attached there. Okay, and plasmalogen has got a protective function against reactive oxygen species molecule. Coming to cardiolipin. So cardiolipin, it is present in mitochondrial membrane. It's a major lipid present in the mitochondrial membrane, and also it is essential for mitochondrial function. So the cardiolipin looks something like this. So it has got a glycerol with two fatty acids attached and a phosphate attached. And so another glycerol molecule with two fatty acid and a phosphate attached, and that is further attached with the glycerol molecule. So basically it is a diphosphatidylglycerol glycerol molecule which is referred as cardiolipin. So defect or deficiency in cardiolipin, it will lead to mitochondrial dysfunction. So it is associated with signs of early aging and pathological conditions including heart failure, hypothyroidism, and Barth syndrome, that is cardioskeletal myopathy, can be seen if there is any defect or deficiency in cardiolipin molecules. So this is all about classification of lipids and some of the applied clinical aspects that are associated with classification of lipids. Thank you.